Our next topic is cloning. Now, cloning is a topic which is very important both from your prelims as well as from mains perspective. And all the aspects of cloning that we are going to learn here will be important for you in one way or the other for your examinations. So first of all, what is the meaning of cloning? Cloning in very simple words would mean creating duplicate of something. Creating duplicate of something will be called as cloning. So in terms of biotechnology, when you are trying to create a replica or a duplicate of a given organism, you will say that you are trying to clone a given organism. All right. Now, the picture that you see on your screen, this is the picture of Dolly the sheep. I'm very sure that many of you are already aware of it. Dolly the sheep was the first cloned animal ever. First cloned animal ever, and this is something that happened back in 1997. Now, this was a remarkable event because until then, cloning was a topic which was more of science fiction than reality. You might have seen many movies and many TV shows, etc., where you will see that there's one guy who goes into a box and there's another one who comes out of another box, who's just a replica of the first person that went into the box. Right. So many of these kind of films in English, in Hollywood, and some in Bollywood also were made. If you have seen Shaktiman, you probably would have seen it. So this process is something which was more of a science fiction at that point of time. But with this particular event, the first successful cloning that ever happened, this was something which is a very remarkable event for biotechnology. Because for the first time, we were able to create a replica of a given animal. Now, this is not something that was simple. They were not just simply able to create a clone. It was 277th attempt. 277 attempts for a successful cloning to take place for the first time. So that's why this was remarkable in the scientific history of human beings. Now, what do we mean by cloning? If you talk about the scientific process, what is the meaning of cloning? Meaning of cloning is, or the process of cloning is more like an asexual reproduction. What is asexual reproduction? Where you have just a single parent from where the child or the progeny is born. So if you talk about many of the unicellular organisms, many of the lower organisms, we see the process of asexual reproduction in them. So asexual reproduction is where you have just one parent. All right. In sexual reproduction, you have two parents, one female and one male, and the reproduction happens, and the progeny born out of this kind of reproduction will be very unique. But when it comes to asexual reproduction, the progeny or the child who is born out of asexual reproduction will be similar to the parent, will be almost same as the parent, because there is only one set of genes that they carry. What happens in sexual reproduction is that there is a mother, there is a father, and the genes from both mother and father are carried in the child. So there is a new combination that we see in the child. But when it comes to asexual reproduction, what we see is that all the genes from the parent is carried forward in the child. So that's why the child also contains the same set of DNA that the parent contains. So this is what happens in cloning as well, that it is a kind of asexual reproduction for all practical purposes. So if you see here, it is a process of asexual reproduction in which the offspring or the progeny is an exact replica of the single parent who has contributed to the genetic material. Now, when we talk about cloning, the process of cloning, when you talk about animal cloning in India, there have been, apart from Dolly the sheep, there have been other examples in the world. If you talk about India, the first example was Samrupa. Back in 2009, this was the world's first cloned buffalo, and the calf was named as Samrupa. Unfortunately, Samrupa did not live for long. Samrupa succumbed to many internal diseases, infections, and ultimately it succumbed to lung infection just within 5 days after it was born. Then after that, the second attempt was Garima, again a buffalo. This time Garima lived for more than 2 years, but again after 2 years it died. Then after that, there is an example of CIRB Gaurav. So CIRB Gaurav in 2016 was another example of buffalo cloning in India. And then after that in 2020, we saw that 7 clones of M29 Murabul and 1 clone of Hisar Gaurav were created. So this is the latest of all the cloning that have happened in India. Now let's try to understand the process of cloning. In cloning, what we do is, first of all, we perform cloning with the help of somatic cells. Now what is the meaning of somatic cell? Any cell in our body which is not reproductive is called as a somatic cell. That is, every cell in the body apart from the egg cell and the sperm, any cell in the body apart from egg cell and the sperm would be called as somatic cells. So all the non-reproductive cells, alright, all the non-reproductive cells in the body will be called as somatic cells. So cloning is performed by somatic cell nuclear transfer into the enucleated cell. What is the meaning of enucleated? A cell from where nucleus has been removed. Alright, from a cell from where nucleus has been removed will be called as enucleated cell. So cloning is performed by somatic cell nuclear transfer into enucleated egg. That's why it is also sometimes called as somatic cell nuclear transfer technology. So what we are doing is that we are taking a somatic cell. From the somatic cell we are taking the nucleus and we are placing it in an enucleated egg cell. Now let's try to understand why we are doing that. So first of all, this will happen. Then after that, activation of this cell will happen with the help of electric shock. So we produce a lot of cells, we produce a lot of copies of cells, and then after that it will be put inside a female, a reproductive female, from where the progeny can be born. An offspring will be born from here. Alright. So within a week, this single cell becomes a ball mass having around 150 unspecialized cells. This stage of development is called as blastocyst. Alright. Now let's try to understand what do we mean. First of all, we isolate the somatic cell. Alright. We isolate the somatic cell. So somatic cell is what kind of a cell? Any cell in our body which is not reproductive. Any non-reproductive cell in the body is called as somatic cell. Alright. So somatic cell first of all will be isolated. From there, what we do is, this nucleus is taken away. This nucleus of the somatic cell will be taken away. Now we take the egg cell. We take the egg cell. From the egg cell we completely take away the nucleus. We take away the nucleus. So you have the enucleated cell. In this enucleated cell, now the somatic nucleus will be placed. So that's why now you have the egg cell with somatic nucleus. Alright. So this, after this, activation with the help of shock will be done, and then early embryonic development will start, where you will create a lot of copies of this cell, and from there are two ways in which cloning can be carried out. Either it will be reproductive cloning or it will be therapeutic cloning. Reproductive cloning, where you want an offspring. Alright, where you want to produce an offspring, it will be called as reproductive cloning. If not, if you just want to store these cells and use it for therapeutic purposes, maybe as let's say for stem cell therapy for example, then in that case it is called as therapeutic cloning. Let's try to understand the entire process again and why we are doing this. First of all, we isolate the somatic cell. From this cell we take away the nucleus. Alright, from this cell we take away the nucleus. This nucleus now will be placed inside the egg cell. Alright, we will take the egg cell from the female, we'll remove the nucleus from there and place this somatic nucleus in the egg cell. And then after that, activation of this cell will happen with the help of shock, and then we'll create a lot of copies of it when the embryonic stage is reached. From there on, either these cells will be placed inside a mother or they will be taken and they will be stored in the laboratory. If they have been placed inside the mother for reproduction to happen or the development of the fetus to happen, then in that case it is reproductive cloning. If not, if it has been stored in the lab, then in that case it is called as therapeutic cloning. Now, one thing, why are we removing the nucleus of this egg cell? What is the purpose? Why is removing the nucleus so important? Now, understand this. Normally, if let's say it was sexual reproduction. In sexual reproduction, what would have happened is that this egg cell would have come in contact with the sperm cell from the male, and the sperm cell and the egg cell would have fertilized together, and thus fetus would be formed from there. So fertilization between the egg cell and the sperm will happen. Now, when you talk about nucleus is very, very important, because nucleus contains all the information of a given individual. So if you have removed the nucleus, alright, if you have removed the nucleus, then the child who will be born, the child will contain only the information from one single nucleus, which is coming from the somatic cell. Alright, the child will contain only one nucleus, and this nucleus is coming from the somatic cell of the parent. Now this parent can be male, can be female, understand this. In sexual reproduction, you would have had one male and one female. Nucleus of both would have come in contact, and the child or the offspring coming out of it would have characteristics coming from both the nuclei, the father's nuclei and the mother's nuclei. But here what you have done is you have taken away the nucleus from the mother and you have placed the somatic nucleus here. So that's why the child who is born out of it, the child will have the characteristics only of one parent, which is coming from the nucleus of the somatic cell. So just remember this, that nucleus contains all the characteristics of an individual. So that's why nucleus is so important, because nucleus contains the DNA basically. It contains the DNA, and this DNA contains all the characteristics of an individual. So that's why if you have removed the nucleus from the egg cell, you have taken away the characteristics of this individual. So that's why all the characteristics will only and only come from the somatic nucleus. So this is the reason why we remove the nucleus of the egg cell. Then after that, activation will happen, and then there are two ways in which cloning can be taken place. Either it will be reproductive cloning or it will be therapeutic cloning. So reproductive cloning in humans right now is not allowed. Reproductive cloning in humans right now is not allowed in the entire world by no government at all, because we don't know in what direction it can go. So right now the only kind of reproductive cloning that is allowed in other animals. Animals like bulls or buffaloes or sheep, we have allowed it, or the governments have allowed it in these animals, but in humans right now it is not allowed. Therapeutic cloning, for therapeutic cloning we do take out the cells like this and we do store it so that it can be used for medical purposes. So in this manner you have two types of cloning. So in reproductive cloning, the newly created embryo is placed back into the uterus, where it takes time to develop into an individual. Reproductive cloning is the production of genetic duplicate of an existing organism. Genetic duplicate of an existing organism. Therapeutic cloning involves the replication of human embryos in order to harvest stem cells for medical uses. In therapeutic cloning, an embryo is created in a similar manner, but the resultant clone are stored in the lab. They are not implanted into the female's uterus. Alright, so they are not implanted into the female's uterus. So that's why they will be kept in the lab, and in case of reproductive cloning, the reproduction will be allowed or development of the fetus will be allowed. So this is the basic difference between reproductive cloning and therapeutic cloning. Now when we are talking about cloning, now there is one question that has come in the past. What is the application of somatic cell nuclear transfer technology? Very simple question. Look at the options. Production of biolarvicides. B. Manufacture of biodegradable plastic. Reproductive cloning of animals. Looks correct. Production of organisms free of disease. Again, very simple question. We are talking about reproductive cloning because your hint here is somatic. We are talking about somatic cell nuclear transfer. Why? Because nucleus from the somatic cell is transferred into the enucleated cell. That's why we are calling it as somatic cell nuclear transfer. So this is nothing but cloning, and one kind of cloning could be reproductive cloning. So that's why the correct answer here will be C. Now there have been concerns that have been raised over human cloning. When I say that human cloning has been banned or hasn't been allowed by any government in the world, there is a strong reason behind it. What are the reasons? Think of the reasons. The first reason could be that it will undermine society's respect for human life. Correct? What may start to happen or what may become a rule would be that people start producing clones. Correct? People start producing clones for various reasons. Maybe you need a house help. Maybe you want to create an army. So there are n number of things that you can think of for which you would create clones. So this will become a major issue, that creation of clones may start to happen, and society's respect for human life will decrease, because now we are able to act God. We are able to create clones. So that's why the respect for human life will not be the same as it is right now. So that's why the entire fabric of society will be completely different. We might start to look at a kind of, let's say, a racial discrimination as well, that clones will be seen as secondary race compared to the humans from where the clones would have been born. So that's why these kind of issues may start to happen. Then after that, it may affect social institutions such as marriage and family. Correct? Then also, children born out of reproductive cloning may be treated as secondary in the family. If we know that, okay, this child has been born out of reproductive cloning, perhaps the respect for this child would not be the same as others. So that's another problem that we might have to face. Then after that, it may create global security concerns, that we might start to see that terrorist organizations, or let's say even armies of different countries, they are starting to create cloned armies, that they are creating clones only for one specific reason, and that is to create an army of their own. They will just be trained for combat and nothing else, because anyway, as we discussed, that the respect for this kind of a life will probably not be the same as compared to a normal human who's born. So that's why these kind of concerns will be there. Apart from that, the concept of designer babies may start to get promoted. What is the meaning of designer babies? I need certain characteristics in my child. I need my child to be like Einstein. Maybe I need my child to look like a film actor. There could be a number of things that I may think of if I'm thinking about my child. Or maybe I want a mix of all these properties. So here again, we will look into a scenario where the parents may be choosing that where do they want the somatic cell to come from. So this is another problem that the society may start to see. And this concept is called as designer babies, because you are designing the baby, or you're designing the child who is not yet born, or you are deciding what kind of child should be born. So that's why this is another problem that may happen with human cloning, that we may start to pick and choose the somatic cell from where we want the child to be born. Then after that, there is another point, that the cloning work actually requires work on embryos. It requires work on embryos, meaning that you will be doing all the research and experiment on embryos. And this is also not correct, because for many cultures, what they believe is that life begins at conception. It does not begin after somebody is born, but rather just after fertilization, when the development of the fetus starts to happen. This is where it is believed that life also begins. So that's why if you are doing research on embryos, if you are killing the embryos, then in that case you are killing a life. And this is also not acceptable for many of the cultures. So that's another problem that we have. All these are various concerns that we have seen with cloning. There's another concern that is raised, that the life expectancy of clones has been observed to be lesser than normal organisms of the same species. Correct? So this is something that we discussed also. For example, in case of Samrupa just lived for 5 days. Even if you talk about Garima also lived for 2 years, but normal life expectancy will be more than 12 to 15 years. So that's why we have seen this as another problem, that the clones which are born out of the process of cloning, the animals which are born out of cloning, they haven't been able to live for long, they haven't been as stable as the normal organism of the same species. So this is another problem that we have faced. Now sometimes there is a confusion that, is there a similarity between surrogacy and reproductive cloning? The answer is no. By the process and the very concept of surrogacy is very, very different as compared to reproductive cloning. Because first of all, in reproductive cloning, we are saying that it is born out of a single parent, correct? Whereas when you talk about surrogacy, surrogacy still is a form of sexual reproduction. So that's why in the basics itself, you cannot compare surrogacy with reproductive cloning. So first of all, in case of process, in process we know that somatic cell is taken for reproductive cloning, whereas when it comes to in vitro fertilization or test tube babies or surrogacy, whatever name you want to give it, in this case we know that the reproduction still is sexual, just that the process is a little different. It is not the natural process. So here also there will be fertilization between the egg cell and the sperm cell, and it will again take place inside a mother's womb. So the process is more or less the same as normal sexual reproduction. So in vitro fertilization or a test tube baby, an egg fertilized by sperm is transferred into the uterus. It creates a progeny similar to the normal conception. The child carries genes of both the mother and the father. Correct? So this is what happens in surrogacy. So that's why surrogacy still is sexual reproduction, whereas reproductive cloning is asexual reproduction. Then after that, uniqueness of progeny. Now, since reproductive cloning is coming from the same parent and only one set of genes it carries, that's why it will be exact replica of the organism from where this clone has been produced. Whereas when it comes to surrogacy, a unique child would be born who would have the genes of both parents, that is mother as well as the father. Then in case of lifespan, we already discussed that in case of reproductive cloning, the lifespan of the clones have been seen as being very less, whereas when you compare it with surrogacy or the child born out of surrogacy is a normal child and will have a normal lifespan as well. And then there are ethical issues. We also discussed about these issues when it comes to reproductive cloning, but issues also exist with regards to surrogacy, and that's why we also have seen that in December 2021 a new bill was also brought in for regulating all the cases of surrogacy in India. Although it has been under criticism because of various reasons, but at the same time there is a new bill that has been tabled into the parliament in December 2021. So that's why, because ethical issues do remain with regards to surrogacy as well. So for example, if you look at this, both in vitro fertilization and reproductive cloning help infertile couples and also same-sex couples with their parenting rights. And this is one of the criticisms actually as well. If you go through the new bill that was tabled into the parliament and was also passed by both Rajya Sabha as well as the Lok Sabha, one of the strong opposition to this particular surrogacy law was that it has not taken into account the same-sex couples, because there is no provision for the same-sex couples also to adopt children with the help of surrogacy. So this is a problem with the law, and that's why it has come under severe criticisms as well. So, but that's another topic, so we are not going to go into the details of it. But yes, this is one of the problems that have been listed out as opposition to the bill that was tabled or passed by both Rajya Sabha as well as Lok Sabha. So these are some of the issues that remain, or these are some of the differences that are there between reproductive cloning and surrogacy. Alright, so these are some of the areas which are important from cloning point of view, and remember that this is a very, very important topic both from prelims as well as from your mains perspective.